Hello, everyone, and welcome to our first spring online event. On this event, we'll be focusing on COVID variants and vaccines. Our special guest today, it's also our longtime member of the Guild, Clemencia Colmenares. She will lead a presentation and group discussion on she will give us an update on basically on the status of the virus, of the vaccines, and post-vaccination safety. Most importantly, today's event will be more a group discussion. Clemencia will welcome you to ask questions during the presentation. She will be talking for about 45 minutes. So if you have any questions related to the topic of the presentation, feel free to unmute yourself and ask the question. If the question is somehow not related with the content of Clemencia's presentation, hold your question until the very end of the event, okay?
I should say, if you want to jump in and ask something and it requires an explanation that's a little lengthy, I might delay it until later. But in general, I think it'll be a lot livelier and a lot more fun if we can all talk about whatever it is we think. And I guarantee if you've thought of a question, even if you don't think it's that important, more than half the rest of the audience is probably thinking it as well. So please.
Yeah, I'm sure everyone has a lot of questions, in particular right now that we are seeing the light at the end of the tunnel. It seems that everything is going back to normal, very slowly back to normal. We will see, maybe Clemencia will tell us how we can get back to normal. Maybe we'll have to be a little bit patient. But we have seen for sure some tour buses already going around the city, so that's an interesting and a positive sign, right? Most importantly, Clemencia, we all know Clemencia as a tour guide and director, but she has done research in molecular genetics for about 30 years, and she also holds a doctorate degree in immunology and virology, so she's like an expert to talk about this topic. So we are in good hands today. And Clemencia, if you are ready, take it away and start your presentation.
Okay, sounds good. Hello, everyone. It's lovely to see you guys again. Okay, other than encouraging you to please not be embarrassed to jump in, I'm gonna go ahead and get started. As someone pointed out to me, people could be mighty sick of this topic and of this damn drawing of the virus, but it's actually a pretty accurate drawing, and so I thought it would be appropriate. The one down, let's see, down here, that's funny, it's not, there we go. The one down here on the bottom right is actually an electron microscope photograph of the virus particles that you see drawn schematically here. So it's pretty accurate. All right, so I've organized this talk starting with the punchline, which really should be located at the end. I organized the talk in such a way that I would explain to you a little bit about what you need to know about the viral genome and the spike protein and how infection works, and then how the vaccines target the spike protein, so that at the end, this punchline would be a little bit more convincing and make a little more sense. But since this is the biggest focus for all of us, how can we protect ourselves from the variants that seem to be arising? I thought I would start from there and then work my way backwards. Okay, so it seems like a no-brainer, but it might as well go ahead and say it. The best way to protect ourselves, it seems to me, and to almost everybody that knows anything about the science, the first thing to do is to get the vaccine. Okay, first of all, protection by the vaccine seems to be greater than the 95% or so that the various studies have shown. In a small study in the US, there were only 3 cases of disease in a group of 2,400 people. So that's less than one, in fact, a lot less than 1%. It's 1 person out of 800. And all of the vaccines, whether, you know, the Moderna, Pfizer, or the AstraZeneca, or the Johnson, all of them seem to be 100% efficient in protecting against severe disease, hospitalization, and death. And I don't know about you, but as far as I'm concerned, that's the bottom line, right? I'm willing to have a cold, even if it gets kind of nasty, as long as I know that I'm not going to end up in the hospital or dying, or cause one of my loved ones to end up in the hospital or dying. Now, the big question is, what happens with the variants? We've all heard that some of the variants may be somewhat resistant. I fully believe, and a whole lot of virologists agree with me from what I can tell from reading out there, that the vaccine will protect even against the variants. That's a likelihood, not a certainty. Nothing in biology is a certainty, nothing is 100%. But once I go through the details of what the spike protein is like and what the variants and the mutations in the variants are like, I think that you'll have more reason to agree with me. But we will save the details of that for later. Okay, so what other ways can we protect ourselves? Sadly, there's nothing revolutionary and new aside from the vaccine. The key thing about this virus is aerosol transmission. So in order to protect against that, what we want to do is avoid large groups of strangers whose behavior we can't absolutely rely on, especially if you're indoors, especially if it's for a long period of time. Sadly, of course, that describes tour buses. So what if you're in a tour bus? I'm here really excited because I have a possible gig at the end of April. And, you know, I'm a little worried. I will do it because I am vaccinated. But what steps will I personally take to protect myself? Okay, it's a group of 15 people, 15. And they're getting us a large bus, so we can have people distancing. They are requiring that they all wear masks, which is good, but I will be wearing a mask, and I will certainly not be wearing just a surgical mask or a cloth mask. I will be wearing either one of my precious N95s or a KN95 that I have bought recently. And I'll go into that in the next slide. The masks, you know, you could run into a little bit of an expense, but I recycle my masks. In fact, I've set up a whole mask rotation system in my living room from chair to chair. And I just, when I take off a mask, I let it sit for 3 days before reusing it. That should destroy any virus. And I do that, I used to do that with my mail and, you know, anything that other people had touched. Thankfully, now that I'm vaccinated, I don't need to do it again. And then the other thing is, as soon as you remove your masks or your mask, disinfect your hands. And I don't use an antibacterial. I just use a simple little spray bottle with some kind of alcohol, either ethanol or isopropanol. And the goal here, of course, is to disinfect your hands before you get any chance to touch your mouth, your nose, your face. And any virus that might be on the surface, you would then transfer to your mouth, nose, and face. So nothing new, but those are the same precautions we've been taking. Okay, you might wonder why a KN95. This diagram is from the CDC. It was what was recommended when medical personnel had to deal with a patient with influenza. which is comparably infectious to COVID-2, coronavirus 2, I'm sorry. And for close contact, they were recommending a surgical mask here, but for any procedures that might generate aerosols, they recommended the N95s. And we know that COVID-2 is transmitted by aerosols. That has been pretty conclusively proven. So this is the mask that I wear, that I purchased. Unfortunately, there are a lot of fake KN95s. I do have some actual N95s that were left over from the wildfires of 3 years ago. So those are my precious masks. These days, I mostly wear these. And they protect against aerosols because they seal completely around your face. It's important to pinch the nose so that it seals completely. And especially for men, if you wear beards, like Roberto, I recommended that he shave it if he has to deal with a group of tourists, because the hair will interfere with the seal of the mask against your face. So, as I said, there are a lot of fake KN95s. You have to watch out for them. These are the websites where you can check whether the KN95 you're dealing with has been approved. And I can email this to anybody who's interested. This is just a slide that showed transmission. You're probably all familiar with it. The problem of pre-symptomatic transmission, which is fairly unique to this virus. But then this transmission by largest respiratory droplets, that's the main route that the virus takes to infect people. Transmission via surfaces, which is called fomites. That happens, but seems to be rare. We're lucky in that respect. One of the things I worry about is that some of the variants out there could mutate and make the virus easier to transmit in this way. But the two really obnoxious, dangerous methods of transmission are by aerosols. And those include both aerosols generated by talking and, you know, heavy breathing when you're running, for example, or exercising, and also aerosols generated when a bathroom is used and the toilet is flushed, which is somewhat grossly called the fecal oral route, but it's very accurate. So in those two circumstances, the only thing that really will protect you is a KN95 or an N95. Okay. Any comments, questions so far? Since that is the bottom line where we stand right now? Nothing? Okay. So I wanted to go back and review some of the basics. So the first thing is the comparison between bacteria and viruses. This is just a size comparison, and it's one of the main differences between bacteria and normal eukaryotic cells and viruses. Here in yellow, you see the size of an average bacterium. Okay, this box in brown is the size of a normal cell from humans or chickens or plants. And the nucleus is about, it's a comparable size to a bacterium. A virus is way, way, way smaller. There are larger and smaller viruses, but in general, a virus is much smaller. So a virus this size would easily go through the pores in a mask. So why does a mask protect us? Well, a virus is not really considered alive. It doesn't have a cell wall that bacteria have to protect them. and it doesn't really metabolize, it doesn't carry out any processes, so it cannot tolerate being dried out. It absolutely requires being in some sort of moist liquid medium. And so what a mask blocks is the minute liquid particles, including the aerosols, that carry the virus. Okay. So this just recaps what I just said. Viruses cannot survive in a dry environment. These are some major differences between viruses and bacteria. It's a lot of information. I don't want to go over all of it. The main characteristics that I think are most important is that viruses are not capable of independent reproduction, whereas bacteria are. And so what do the viruses do to reproduce? What they basically do is they take over the host cell, hijack it, and force it to produce viruses on their own. Whereas bacteria go through a normal process of fission and cell division, copy their own DNA, metabolize, and then divide. Okay, there we go. Okay, so just to recap very briefly, what happens after a virus infects the cell? I'm not going to talk about when and how a virus infects the cell. That I'm saving for when we talk about the spike protein in a couple slides. But right after a virus infects a cell, the virus has just one goal, and that is to reproduce, right? Just like all living things. So it has to hijack the cellular machinery. So some of the viral proteins that are made inside the cell take over all of the viral cells, okay? Force the cell to produce just the viral RNA and just the viral proteins, and eventually to package the virus into a little viral particle. And a single infected cell can make thousands of them. Sometimes during the process, the cells are killed. In the case of COVID-2, it seems like they're usually not killed, although that's not exactly clear. There are some viruses that kill most of the cells they infect. COVID-2 doesn't seem to be one of those. So the immune response comes in and tries to defend against the virus. In the case of COVID, the immune response seems to be both the defense. Early immune response interferon is very important for the early response, early defense, but it also seems to be the cause of a lot of the damage. I wasn't planning to get into more of that. If anyone wants to discuss it, we can talk about it later. But if we want to talk about the variants, I have to review a little bit about the virus's genetic code. So just to go back, the genetic material is usually DNA. With viruses, it's either RNA or DNA. Here we see a double-stranded molecule, so that is by definition DNA. And DNA carries information very similar to a computer code, except instead of being just ones and zeros like a computer, it has 4 symbols, A, T, G, and C. So it can get a lot more complicated than a computer can, actually, hard as that is to believe. Now, in the case of the virus, we are talking about RNA, and so instead of T's, which are thymines, the virus has uracils, and it is single-stranded. So instead of this double one, pretend that, you know, this yellow strand is gone and there's only a single strand. Okay, but otherwise the nucleic acid behaves very similarly. Okay, so there is actual information in the sequence of these bases or letters, and the information codes for amino acids. And 3 of the bases code for one amino acid, pardon me. These amino acids get hooked up one to another, and they make up chains of proteins. Okay, now the proteins can be incredibly short, you know, as little as 5, in which case they're called peptides, but that's just a silly comment, a silly terminology, or they can be thousands and thousands of amino acids long, okay? So then, and the important thing to remember is that we say that there can be a mutation in a gene, but that mutation will have no effect unless it's translated into the protein, and proteins do all of the work. They make cells reproduce, they make viruses reproduce, they do all the metabolism, they make your skin and your hair and your eyes the color they are, they make your liver be a liver instead of a lung, all of that, all of it's done by proteins. Okay, and so proteins, which start out as chains, long or short chains of amino acids, then because of chemical interactions between the molecules in the amino acids, they fold up into all sorts of infinite numbers of structures, and they can assemble into complexes of more than one of the same protein. And I bring that up because, as you'll see, that's the case for the spike protein. Okay, any question there? Have I left anybody behind? Please jump in.
So
Okay.
Clemencia,
Don't be shy. Please ask questions to Clemencia.
Yeah, okay, somebody had a question?
yeah, I've got a question, and there's a request. You might be able to flash back real quickly to the slide that you had for the KN95 quality verification. And a couple of people might want to get a screenshot or get that address now. Saves you answering.
Yeah, why? Well,
There you
it'll be on the recording, right? But it's probably too small here. I'll very happy to send you or help you with locating this because it can be tricky. But this has a list of all of the companies and all of the approval numbers. And the approval numbers are found, let's see. I'm having trouble.
That's okay. That's okay. The choice is either get a screenshot like just now, or get a screenshot from the YouTube video that'll be up by Monday at the latest.
Yeah, but also email me. I have this all the time. So this is the important part on the mask. It has the approval number and the company number, and it should say NIOSH. I have found one mask that appears to be legitimate that doesn't say NIOSH, but and the other detail to remember is there are KN95s out there sold that have ear loops. There are no legitimate KN95s with ear loops. All of those are fake. All approved KN95s go around your head. So just keep that in mind.
Well, just start talking, see if you can.
Yeah, go ahead. Nancy?
Oh, yes, you had a
I was
question.
going to ask a question that goes back a little bit. When you say the vaccines are 95% effective, I'm trying to understand this in my simple way, and I probably got it wrong. Does it mean that if you take 1,000 people who were selected, who were COVID negative, and were given the vaccine, and then over a period of time, months, whatever, were tested to find out how many of them either became positive, became ill, or in fact, unfortunately died, and then compared that to 1,000 people who were tested negative, but over that same period were not given the vaccine?
Correct.
How would they compare?
Okay, so if it were 95%, then 50 people out of those 1,000 would have gotten disease.
Yes. And what about the
Okay.
people who were selected were negative and were not given the vaccine over that same period of time, and then assessed at the end of that period, the same period? Has that test been done?
Yes, yeah, that is exactly the test that was done. That was the trial.
But what was the average result of those people? How many of those people either tested positive, became ill, or in fact died? I mean, comparing to the vaccinated group.
Yeah, okay. I don't remember who died, but as I recall, it was it was 5%. So the actual numbers that gave us the 5% were like 8 people in the first very smaller group, 8 people who got sick after having received the vaccine,
Yeah.
versus like 180 people who got sick.
So they selected, of the people who were selected and observed over the same period of time, who had not gotten the vaccine and were negative when they began.
Correct.
180 versus 8 people were, and this has been repeated. This is this, and this.
Oh yeah, similar results. Yeah, for Moderna and Pfizer, the numbers are very similar, and the study was done ultimately with 20,000 people. So the numbers are really, really outstanding.
So out of the 20,000 people, 180 versus 8. Is that what you're saying?
Maybe what? I'm sorry, I didn't hear you.
Out of 120,000 people, or 20,000, you said?
No, 20,000.
So out of 20,000, 8 of the vaccinated people became ill or became positive, I'm not sure. And
well, so 8 is the initial group. Of the whole 20,000, it was a little bit more, but boy, it was not a lot. Yeah, sadly, I don't remember the exact numbers, but I can email you the data. But to me, the more impressive statistic is in that whole 20,000 group. And then there was another group of like 22,000 for the Moderna vaccine, and then comparable groups for the AstraZeneca and the Johnson. The protection there against severe disease, hospitalization, and death is, as far as I can tell, 100%. So none of the people that received the vaccine got severely ill, went into the hospital, or died. None of them.
So of the 20,000 individuals, I want to end this, but I'm just saying, for the 20, because this has been dogging me, of the 20,000 who didn't get the vaccine, who were selected initially, I presume, to have been negative,
right?
You're saying that 180 of them got sick, or became positive, not necessarily got sick. This seems to be the statistic that's important to evaluate the claim of 95%.
Right. So when you expand it to the 20,000, I think it was more than 180. Sadly, I wish, I just don't remember the exact number. I'm sorry, and I don't want to pull something out of a hat, but I'm pretty sure it was more than 180. But yeah, and out of those, I want to say that ultimately out of the 20,000, there were maybe between 50 and 100 people who got severely ill, were hospitalized or died. You know.
And out of the vaccinated group, only 8. Or how many of those 8 were severely?
None of them. That's the whole thing. None of them. There was 0.
There was 0 out of. Okay, that's what I wanted to know. So out of the test group and the group that wasn't given, only 8 individuals became what? Ill or
something else?
Moderately ill. Yeah, maybe we can keep talking about this later.
Sorry to take up time with this. Thank you. I appreciate your response. I've been wanting to find out about this.
Oh, sure. You're welcome. Yeah, I mean, I can look it up and give you the exact numbers. I just don't remember them right now.
Clemencia, can I ask how long a vaccine provides immunity?
Okay, yeah, that's a really good question. We don't know that so far because, of course, the test is still going on. Easily 6 months at least. Some people are guessing at least a year. Some people are guessing at least a couple years. Given the variants, I am guessing that they will come out with a booster that includes better protection against the variants, and they'll probably target that for about a year. So for a little while, you know, the next year or 2, we may have to have another booster, kind of like influenza. but at least 6 months is what they've found out so far.
Okay, and maybe that's why the UK health minister says that people might need a COVID vaccine every 6 months. Is that indefinitely, or is that just for the first year or so until we get to herd immunity?
I have no idea why he's saying that, other than 6 months is just the period that they've had right now to look at since the first vaccines were given. So he's being just incredibly restrictive and by the letter. You know, they just don't have any information of beyond 6 months so far.
So there's potential that it could be more than 6 months, but that would be
yeah.
the minimum.
Oh yeah, absolutely.
Thank you.
Sure. Okay, so we had gone this far about, you know, proteins folding up into all sorts of structures. Now, so I hope it's clear why, when we talk about the structure of a genome, a nucleic acid sequence, or of a protein, we usually refer to it as a straight line. Okay, and the straight line just refers to this sequence of amino acids or nucleic acids. Okay, so this is a schematic of the entire almost 30,000 bases of the COVID genome. Okay, by far the biggest proteins are these two large ones, and these are involved in copying the virus RNA. But this little bit here that I have circled is the spike protein, the famous spike protein. And that's the protein that's most important for infection. Okay, now out of the nearly 30,000 bases of RNA, the spike protein is 3,819, or almost a tenth. Okay, and that gets translated into, again, a linear sequence shown here of 1,273 amino acids. So this represents all of the spike protein. And of course, the spike protein has different parts, okay? And I'm not gonna, you know, really describe the names of the parts. The only thing that's really important is this part in green right here, because that's the part of the protein that encodes the receptor binding domain, meaning that's the part that sticks to a receptor on human cells, which is the ACE2 receptor. And that's how the protein, the spike protein, infects cells, okay? Here is a more drawing-like representation. Here, these structures that stick way out of the virus are representing the spike protein, which, by the way, is a trimer. It's a threesome. 3 of the same spike molecule collect to make a spike protein. We'll go into that in a little more detail in a little while. And of course, this viral particle I told you was tiny. So here we're showing it hugely, hugely enlarged many times, okay? And the end of the spike protein is the binding domain, and it sticks more or less tightly, depending on the variant, to the ACE2 receptor, which is a molecule that sticks out of the cell membrane. That's shown here as a double layer lipid envelope. By the way, that's the same lipid envelope that is found in the virus. Okay. Now, oh, I forget. Okay, so that's the very first step that has to happen for infection, is this spike protein has to bind to the ACE2. So I'm going to show you a couple of images of a variant, the Brazilian variant. But to do that, it's a lot easier to show you the whole viral genome. It's still a continuous line, but if you bend it into a circle, it makes it a lot easier to show you all of the mutations, and you'll see why in the next slide. So here's the genome. Here's the two big first proteins that are involved in copying the virus. And here is the spike protein, which is about 1/10 of the viral genome. This is the stuff we've already seen. And it's important to remember here that the virus gets copied, and every time it gets copied, there are mistakes made. Every time our cells copy DNA, there are mistakes made. That's just inescapable, even though there are correction mechanisms. And those mistakes happen at a constant rate. It's a very low rate, but it's constant. And the more times you copy it, then the more chances you have of having different mistakes made. And the more times, kind of like if you have enough monkeys typing randomly, one of them will come up with Shakespeare. Well, enough mutations that happen increase the chances that you're going to come up with something worse and worse, right? So let me show you the Brazilian variant in this kind of a diagram. So the Brazilian variant has just a few changes here at these spots you see. And by the way, the nomenclature of these changes is very simple. It's the original amino acid followed by the position of that amino acid, followed by what that amino acid changes into. So it's very simple-minded. But you'll notice that the Brazilian variant really doesn't have very many mutations until you get to the spike protein. And then it has a lot of mutations. And of course, in the spike protein, a lot of those mutations are concentrated in this area, which is the receptor binding domain. Why is that? Well, because that's the part that's going to infect cells. And so if you got a virus that has a mutation there that makes it better at infecting cells, it's going to be more successful in transmitting itself to new people and new generations. I've also pointed out one mutation here called N501Y, which is shared with the British variant and the South African variant. So that's a really big clue. Those are also somewhat more infectious viruses. So that's a really big clue that that particular change of amino acid N or asparagine to a tyrosine at position 501 of the spike protein is involved in producing more successful viruses, right? We see it happening over and over again in a more infectious virus. It's just a matter of numbers and probability. Okay, sadly, this figure is a little older, so it doesn't show the more recent changes. Sorry. But when it was published, it showed all of the different changes that had been observed to date in the spike protein. And as you can see, this is the whole spike protein, and there are some changes in other domains, but the vast majority of them are concentrated right here in the receptor binding domain. And also the color coding is such that blue means that it's more infectious, yellow means that it's less infectious, and pink means that it's more sensitive to monoclonal antibodies. Damn it, I have trouble with this. And green means that it's less sensitive to monoclonal antibodies. So you can see that there have been lots and lots of mutations. One of the interesting things to me here is that actually the mutations that caused increased infectiousness are actually fewer than the yellow mutations, which are the dominant number by far. So this just illustrates the fact that the virus is always changing, and it's a process that it's not really, you can't change it. In fact, we probably have very little idea of what kind of variants are arising in third world countries where the virus isn't sequenced. So we just hope that something really nasty doesn't arise out there. And sadly, the more viruses that reproduce, the more and more of a chance there is, of course. Okay, now let's talk about the vaccines, which are directly related to the spike protein sequences. So it should be clear from what I've said so far why most of the vaccines out there have focused on the spike protein, right? Because it's the key to infection. It attaches to cells, and the RNA, the receptor binding domain is key to infection. So this is a diagram of what the more traditional, the older style of vaccine used to be like, okay? And basically, it shows, intact virus particles. And the old vaccines used various different ways, either radiation or chemicals, of either crippling or killing the viruses. It should be very clear that this method is somewhat risky, and what it requires is absolutely excellent, fantastic quality control, right? Because you don't want a process that's going to leave some viruses uncrippled or unkilled. And so the major example of this, there's also an empty virus shell, but that's actually a much harder process. So the main example of this is the Sinopharm Chinese vaccine. They use, I believe it's a crippled virus. And, I consider it somewhat risky. I have heard just anecdotal reports of people who have gotten COVID after getting this vaccine. I don't know if they're true or not, but certainly the method of preparing the vaccine suggests that it is possible. So I would actually refuse this vaccine if it were the only thing that were offered out there. Okay, more recent vaccines express or take the approach of expressing a viral protein from a fake virus. And in this case, the virus that's been used far and wide is an adenovirus. And of course, they have chosen the spike gene for reasons we've already seen. And examples of this are the Russian vaccine and the AstraZeneca vaccine. Okay, the problems with these this approach are that you can do a booster. The AstraZeneca tried first one vaccine and then a booster, and it turned out that a second dose helped. But more boosters get less and less effective because your body develops immunity not just against the spike protein, but against the other components of the adenovirus. And so it doesn't allow the spike protein to be expressed efficiently once you're immune, because it blocks the virus from infecting your cells, the fake virus, which is what you need to develop immunity. So I hope that's clear. So for example, if you want a booster against variants later on, the adenovirus vaccines would probably not be the greatest choice. Okay, and then the newest type.
So if your body builds up an immunity to the adenovirus, does it at the same time build up an immunity to the coronavirus, or does that?
Not to the coronavirus. Yes, to the spike protein, which is expressed within the adenovirus. It's a molecular construct. So they've synthesized the spike gene and stuck it inside this fake adenovirus that's produced in the lab.
But if the resistance is to the spike protein, then would that confer some resistance to the coronavirus also?
Yes, yeah, absolutely. That's why the vaccines are effective. I keep having trouble with this, sorry. Yeah, so the spike protein is expressed as part of the adenovirus. So yes, you get immunity against the spike protein, which is why you are protected against the coronavirus. But then if you would try to express a different spike version, the one that's produced by the variants, it would be less successful because your body would attack the adenovirus and not allow it to express the new variant spike protein. And so you wouldn't develop as much immunity against the new spike protein. Does that make it any clearer, Andy?
Yeah, thank you.
Sure. Any other questions about this? I know my sister used to get freaked out when I would tell her that we were making viruses in the lab, but it's something you do all the time. It's Okay, so this illustrates the newest method. And that is one in which we take the spike gene, and there's two possibilities. You could either make it into DNA and then allow the cells to copy that into RNA and then make protein. But so that has serious problems. Turns out that pieces of DNA can integrate at random into the genome, and they can cause a lot of genetic problems, the worst of which would be cancer. Okay, and so, and this is more likely to happen unless the DNA is delivered by viruses, which is why the strategy that's used is the adenovirus. So this is really not a great method. Oh, I'm sorry, I made this 3 and that 4. Okay, my mistake. But so the alternative is to deliver the vaccine as RNA, which is much safer. But the problem here is that RNA is very, very easily degraded for a lot of reasons that I'm sure you don't want to hear about. So to make the vaccine, they had to chemically modify the RNA and make it more stable and wrap it in lipid. And of course, the examples of these as are the Moderna and the Pfizer vaccines. Okay, so that's a summary of the vaccines that are out there. Just to recap, the spike protein gene was copied many, many times over. Come on. And copied into RNA. And then those mRNA molecules, the way they were made, they're chemically modified to protect them against degradation. Then they have to be coated with lipids to further protect the the RNA, sorry, and to enable it to get inside the cell. So, hang on. Okay, this lipid coat so lipids are fats, and fats are the same thing that surround human cells. And so this lipid coat makes it very easy for this fat to kind of merge with the fat that surrounds the cell and gives it a membrane. Okay, and so the mRNA gets delivered very easily into the inside of a human cell. Okay, and this is just my own observation. It seems to me that the lipid coat that Moderna uses compared to the Pfizer is probably a little more stable because the Moderna vaccine doesn't need as deep freezing. All right, so if we put it all together, why do I say that the vaccines, as we've described them so far, should protect at least a little bit against all of the variants? So this image shows you we've gone over why the variant changes that really matter are concentrated in the, not RNA, receptor. that's my mistake. I'm very sorry. Okay, receptor binding domain. Okay, so RBD. So this is a schematic that shows you 3 spike proteins. Here's 3 of these little structures. This stalk shows 3, this S1 shows 3 subunits, and this RBD in blue shows 3 subunits. It turns out that this domain usually sort of sits down against this other S1 unit. And in order to bind to the ACE2 receptor, it has to stand up to become erect, as shown by this arrow. Okay, so this is a schematic drawing. This is actually a little closer to the way the protein structure actually looks. I told you that proteins fold themselves up into all sorts of bizarre shapes. Well, here you see those bizarre shapes, and this actually shows you the threesome. This is 3 spike proteins, 1 in yellow, 1 in blue, and 1 in brown. And this is looking at it from the side, but this is looking at it from the top. And this is what they call the closed configuration, where all of the receptor binding domains are lying down. Then if you go over here, one of them has become erect. And that is what's required for the ACE2 now to bind to it. Okay, so this domain is the most important one for binding to ACE2 and infecting a cell. And we've seen that this domain is where most of the changes that affect infectiousness are located. But remember that the vaccines were designed to express the entire spike protein, not just the receptor binding domain. So when you make antibodies, and very importantly, these vaccines are very good at inducing your body to make T cells against the spike protein as well. So not just antibodies, but also T cells. Antibodies and T cells are made against the whole thing. And so the variants are more likely to have changes here, but your body is reacting against the whole protein. So that gives us a much more improved chance of resisting the virus when it infects. But it should also be clear that the method for making the vaccine, all you have to do is synthesize a new variant type of RNA, which is really easy to do. It's done in the lab in seconds. And the whole other, the wrapping the RNA in lipids, all of that technique, that's exactly the same. Nothing gets changed. It probably doesn't even need any further FDA approval. So that's the really good news, that for boosters, we should be able to generate new ones really, really quickly. So I think I'm gonna stop there and take some more questions. Hopefully I've somewhat convinced you. But are there any more questions?
Feel free to open up your or unmute yourself and pop your question directly to Clemencia. Thank you, Clemencia. Wonderful presentation.
Sure.
Okay.
Yeah, this is Anthony. Thank you. I'm going to shave. I'm going to shave my beard. It's too long.
Yeah, I mean,
Yeah.
I guess it just depends on how worried you are, but I was very worried. I've been wearing KN95s even in grocery stores. And in fact, I'm so happy now because after the vaccine, I don't have to sanitize all my groceries, which I was doing, and it was a huge pain. So,
so tomorrow, tomorrow you can see me, Fisherman War, Saturday, Sunday, and Red and White Fleet is very packed, and everyone, all those restaurants very packed.
Really?
Yes.
Yeah, so that's a little scary because you have to take off your mask to eat. So You know,
And to
I've
drink.
gone to a couple of restaurants, but I wouldn't feel comfortable unless, you know, there were some good social distancing, and I don't know that I'd feel comfortable indoors just yet. I would wait a little while. So, by the way, the other thing to do is, I'm sure that it'll be in the news. You know, they've already said, and it's no surprise that the variants, including the Brazil variant, are now found in the US. Those will probably spread a little bit, but as long as there aren't huge numbers of those in our community, you know, I think it would be okay. Yeah, any other questions? Let's see.
Don Delora, take it away, Don. Pop the question.
It's Ellen. I had a question about the monoclonal antibodies.
Yeah.
Are those harvested from people who have had the disease, or are those now being produced in a lab?
No, okay, those are all produced in a lab, and I think I can explain it relatively easily. If we go here, so a monoclonal antibody is an antibody that recognizes it's a single antibody molecule. And so it recognizes one antibody molecule recognizes a tiny part of this protein. In fact, if this is 1,270 amino acids, one antibody recognizes a part that's about 6 or 7. So you can imagine how many antibodies your whole body might produce. I mean, literally thousands and thousands of combinations of 6 or 7 amino acids here, right? So a monoclonal antibody is always made in a lab. Your body always produces a giant mixture. But that's an excellent question. Thank you. It really clarifies things.
Okay, Nancy Thomas, you're up for a question.
Yes. After your wonderful presentation about the spike protein and the body's reaction, a very basic question. It seems that there are two processes going on here. One is your natural immunity, which responds to the presence of the virus. And then it's your immunity responding to the presence of the spike proteins that are produced by the vaccine. That these are two processes going on at the same time. But that the spike proteins provide an opportunity for more of an attack on the virus than just the virus itself present in your body?
Okay, so your body is not responding to the virus if you haven't been already infected first.
Yeah, but I mean, there's no natural immunity to COVID.
No. They thought that there might be some pre-existing immunity because there are other coronaviruses, not just SARS and MERS, but other, you know, very common cold, if you will, coronaviruses. But that doesn't seem to be, there doesn't seem to be much of that. So no, you don't have any natural immunity. In fact, the only sort of spontaneous immunity would be interferon, which is not specific against coronavirus. It's just, you know, antiviral in general. Now, that's really important. If you don't have a good interferon response, those are the people who get very sick, but it's not virus specific at all. However, your immunity against the spike protein, that is very virus specific. And in fact, it is recommended that people who have had the viral infection, have gotten sick, do get the vaccine anyway, because it does seem to provide better and longer lasting immunity. That's what they think right now.
Well, your body will not respond in an immune efficient way to the spike protein on the actual virus, but it will respond to the spike protein produced by the vaccine.
Well, no, it does respond to the spike protein on the virus. It's just that that response will take too long, and in the meantime, the virus has done a whole lot of damage.
So the spike proteins produced by the vaccine sort of push the body to produce protection at a greater rate than it would normally to the virus itself.
Yes, that does appear to be the case. Yes.
Okay, thank you very much. We're running up against the clock. Thank you for that question. Anybody else got one or two last questions for Clemencia? Clemencia, how?
Yeah, there was a question in the chat about how can a company make money with only 15 passengers? Well, so this is a you know, a private tour group. So I assume they're going to build it in, but yeah, I mean, it's not something that's really sustainable.
Yeah, that's actually, I didn't see that question. Thank you, Clemencia.
Sure. And then the question about Florida. Yeah, I would say that the vaccine is the only way to explain why cases are still dropping. And I mean, I guess the other thing is that the surges were probably due to the holiday get-togethers. So now they're dropping because people were distancing again, but if they've recently removed all mask and distancing restrictions, then if there are in fact resistant variants, we could be seeing further surges. And that's why some virologists and epidemiologists are really worried, because you know it could be getting worse again, but hopefully not.
Well, let's all put our hands together and clap in appreciation for Clemencia's contribution. Thank you all for joining us. And we look forward to future meetings coming down the pipe, and stay tuned for an announcement, email coming out for the posting of the YouTube video.
Thank you, Clemencia. Very good job.
Oh, thanks. Glad you enjoyed it. Good to hear from you, Janice.
Well, I live the life you're living with my family, with the mail and wiping the groceries, and so I feel very safe. And I'm vaccinated as well, so now I know.
Isn't it wonderful? I mean, I feel I feel so happy now when we go shopping. It's so silly.
I'm not agile enough to kick my heels together, but I would if I could.
I agree.
Thank you so much.
Oh, you're very welcome. This was really fun.
Okay, again, thanks, Clemencia.
Take it easy, Len. Oh, yeah. Hey, Len?
Go ahead.
Can I send you the websites for the masks so you can send them to people?
By all means, yeah, do that, and I'll just put it in the announcement that we send out.
Okay, sounds good.
Okay,
all right.
Great. Take care.